In this explainer, we are going to walk through a 2009 paper by Petersen and Reddien that asks a fundamental question in regeneration biology. When a planarian is cut, how does it know whether to regenerate a head or a tail? More specifically, does polarity come from passively rescaling pre-existing positional gradients, or does wounding itself actively trigger a new program that specifies tissue identity? That is the central problem this paper addresses. To organize the story, we will move through 6 sections. First, we will briefly explain Wnt signaling, since it provides the mechanistic foundation for the paper. Second, we will define the polarity problem in planarian regeneration, how a wound knows whether to rebuild a head or a tail. Then, we will examine the first major result, showing that Wnt-P1 behaves as a wound response rather than simply a static posterior marker. From there, we will follow how that finding leads to a broader Wnt signaling cascade involving beta-catenin 1 and Wnt-P2. After that, we will ask whether this polarity setting program is stem cell independent, or whether it is merely a consequence of neoblast-driven regeneration. And finally, we will bring all of these results together into an instructive model for how regeneration polarity is established. So let's get started. Before we get into the experiments, we need a short review of canonical Wnt signaling, because this paper is built around that pathway. How a single cell knows what it's supposed to become is dependent on cell communication. Cells get these communication relays from signaling cascades, one of the most important being Wnt signaling, a conserved pathway that controls cell fate. In its most simplistic way, you can think of this pathway like an on and off switch for the cell. Now let's meet the star of our show, a protein called beta-catenin. Its fate decides everything. Let's start with the default state, off. So this is what's happening inside the cell when there's just no signal around. So inside the cell, you have this group of proteins, axin, APC, CK1, and GSK3 beta, that form a destruction complex. And its job is pretty much what it sounds like. It phosphorylates beta-catenin, marks it for degradation, and keeps it from entering the nucleus. Its levels stay super low. And the result of that? The genes this pathway controls stay silent. The switch is firmly off. But everything changes when a Wnt ligand arrives. So let's see how this switch gets flipped on. Proteins latch onto cell surface receptors like frizzled and LPR5 and 6 and kick off a chain reaction. This action yanks that destruction complex to the membrane, and just like that, the destruction complex can destroy no more. Now that it's safe from destruction, beta-catenin is finally free to build up inside the cell. And here's the big payoff. All that accumulated beta-catenin travels right into the cell's nucleus. Inside, it partners up with other proteins kicking out repressors that were keeping the genes off. Together, they form a new activation complex. The switch is now on, and specific genes are activated in that journey from the cytoplasm to the nucleus. That is the absolute hallmark of this pathway's activation. So to recap, Wnt off equals beta-catenin gets destroyed, and genes stay silent. Wnt on means genes are on. So why does this one little switch matter so much? What's the big deal? It matters because in planarians, canonical Wnt signaling is a core determinant of anterior-posterior identity. It controls nothing less than cell fate. Broadly speaking, posterior Wnt and beta-catenin activity promote tail fate, whereas reducing Wnt signaling promotes head fate. By 2009, previous RNAi studies had already shown that inhibiting beta-catenin 1, or Wnt-P1, a gene of Wnt signaling, could convert posterior wounds into heads, while inhibiting APC, a negative regulator of beta-catenin, could convert anterior wounds into tails. So the field already knew Wnt signaling mattered. The unresolved issue was how that polarity was reestablished after injury. So now that the pathway is on the table, let's shift to the biological system itself. Planarians are remarkable because they can regenerate essentially any missing body region. They do this with the help of adult stem cells called neoblasts, which provide the cellular source of new tissue. In the broader, later framework of the field, those stem cells are guided by positional information, much of which is encoded by position control genes in body wall muscle. But this 2009 paper sits at an earlier mechanistic step. It is asking how the initial polarity decision is made immediately after injury, before stem cells have rebuilt the missing structure. So that sets up the central problem very clearly. Is polarity restored by passive rescaling of pre existing gradients or by active wound induced input? To get at that question, the paper begins with one key gene, WNT-P1. In intact animals, WNT-P1 is expressed only at the posterior pole. If polarity were restored simply by rescaling pre existing gradients, that is roughly what you would expect. Posterior associated genes should remain posterior associated. But the first result is surprising. In Figure 1, the authors took a planarian and surgically cut it into three parts, establishing a head portion, a trunk portion, and a tail portion. The head portion would need to make a trunk and a tail, while the tail would need to make a trunk and a head, and the trunk portion requiring both a tail and head, respectively. Then, they stained for expression of WNT-P1 protein between time of initial cut 0 hours to 24 hours. What they saw was that WNT-P1 is induced at both anterior-facing and posterior-facing wounds within this time span, meaning that the earliest response of WNT-P1 is not restricted to posterior tissue. It is symmetric. And that immediately argues against the idea that WNT-P1 is merely reporting pre-existing posterior identity. However, what happened between 48 and 72 hours made things even more exciting. WNT-P1 became exclusively restricted to the regions that require a tail, while anterior-facing regions tuned down WNT-P1. That immediately raises the next question. Is this induction specific to amputations that require tail regeneration, or is it part of a broader wound response? In figure panels F through J, the authors test several kinds of injuries: slices, punctures, wedge excisions, oblique cuts, and lateral cuts. In every case, WNT-P1 is induced after injury. Only later does the pattern diverge. Anterior wound expression disappears, whereas posterior wound expression persists and consolidates at the posterior pole. So the first major conclusion of the paper is that early WNT-P1 induction is a generic wound response, and the later posterior restriction is a second phase layered on top of that. So already, the paper is pushing away from passive rescaling and toward an active wound-induced program. But observing a wound response is only the beginning. The next question is whether that early induction actually matters functionally. To test that, the authors knocked down WNT-P1 by RNAi. In head fragments, the posterior-facing wound should regenerate a tail. Instead, after WNT-P1 knockdown, some of those posterior wounds regenerate a second head. And the authors do not rely only on appearance. They use positional markers to show that the posterior blastema has truly become anteriorized at the molecular level. In other words, WNT-P1 is not just associated with posterior identity, it is required for it. They strengthen this further with a lateral regeneration assay. In that context, the animal must rebuild tissue along the side of the body, rather than simply making a head at one end and a tail at the other. When WNT-P1 is inhibited in this assay, the regenerated tissue becomes too anterior-like. There is expanded anterior marker expression and formation of extra photoreceptors. That tells us WNT-P1 is not only involved in making tails at posterior wounds, it also helps prevent excessive anteriorization when new tissue is rebuilt along the body axis. At this point, the paper asks an even sharper mechanistic question. Does polarity depend only on pre-existing WNT-P1 protein that was already in the tissue before injury, or is newly produced WNT-P1 after wounding actually required? So, they amputate first and only then deliver WNT-P1 double-stranded RNA inhibition. Even with this post-amputation design, they still get posterior head regeneration. That is a very strong result because it shows that the crucial signal is not just residual protein left over from the intact animal. The system specifically requires new expression of Wnt-P1 during regeneration. With that established, the authors then ask whether another Wnt gene might act together with Wnt-P1. This leads them to Wnt-P2. In a sensitized trunk fragment assay, single knockdown of either Wnt-P1 or Wnt-P2 does not strongly disrupt posterior regeneration in that context. But when they knock down both genes together, posterior-facing wounds regenerate heads instead of tails with high penetrance. That genetic enhancement shows that Wnt-P1 and Wnt-P2 cooperate in posterior specification. Expression timing then helps place Wnt-P2 in the sequence. Unlike Wnt-P1, which is induced at any wound, Wnt-P2 appears only at posterior-facing wounds, and it appears later. That timing matters. It means Wnt-P2 is not a generic injury response. It is a posterior-specific downstream program that comes on after the early wound-induced phase of Wnt-P1. And that brings us to the mechanistic heart of the paper. The authors ask whether beta-catenin 1 is required for the early wound induction of Wnt-P1. The answer is no. Even when beta-catenin 1 is knocked down, early wound-induced Wnt-P1 still appears at both kinds of wounds. That means the initial induction of Wnt-P1 is not triggered by canonical Wnt and beta-catenin signaling. Some other wound-derived signal must be upstream. But when the authors look at Wnt-P2, the logic flips. Knocking down either Wnt-P1 or beta-catenin 1 abolishes normal induction of Wnt-P2 at posterior wounds. So now the hierarchy becomes much clearer. An unknown wound signal induces Wnt-P1. Wnt-P1 then acts through beta-catenin 1, and that induces Wnt-P2 specifically at posterior-facing wounds. At this stage, the paper has assembled a very compelling signaling cascade, but there is still one crucial alternative explanation to exclude. Maybe this Wnt program is not actually instructive. Maybe it is just a consequence of stem cells beginning to regenerate tail tissue. To test that, the authors eliminate neoblasts by irradiating the animals before amputation. This is the key causality experiment. If the Wnt cascade still occurs without neoblasts, then it must be upstream of stem cell-mediated regeneration. And that is exactly what they find. In irradiated animals that lack neoblasts, the early wound-induced Wnt-P1 response still occurs, and Wnt-P2 is still induced in the posterior. What fails is the later posterior pole persistence of Wnt-P1. So the early polarity program is still running even when the animal cannot actually build a new tail. That means the early Wnt program is not a downstream consequence of regeneration. It is an upstream instructive program that tells the system what to regenerate. The stem cells come later and implement that instruction. So with all the major pieces now in place, we can step back and assemble the full model. A wound generates a generic injury signal. That signal induces Wnt-P1 at any wound. At posterior-facing wounds, Wnt-P1 then acts through beta-catenin 1 to induce Wnt-P2, creating a posterior-specific transcriptional state. This polarity setting program happens in differentiated tissues and does not require neoblasts. Only after that decision is made do neoblasts implement tail formation. In other words, the paper replaces a passive gradient rescaling model with an active instructive model of regeneration polarity. And that is really what gives the paper its broader significance. It is not just about one Wnt gene in one flatworm. It shows that regeneration is not only about stem cells and growth. It is also about rapid wound-induced information processing, how an injury is interpreted and translated into positional identity. Later work in the field would show that much of this positional information is encoded by position control genes in muscle, and broader comparative work would show that canonical Wnt signaling remains deeply conserved across planarian species. But the conceptual breakthrough here is already complete. Wounding itself actively participates in resetting the body axis. So the final takeaway is this. In planarian regeneration, polarity is not simply inherited by passively rescaling what was already there. Instead, injury launches a new signaling program. That program begins with a generic wound response, resolves into a posterior specific Wnt cascade, and instructs stem cells what structure to build. And that leaves the field with the next major question. What is the upstream wound signal, and how does it interface with pre existing positional information to produce such a precise outcome? Thank you all very much for taking the time to listen. Please check out the paper in its entirety with the link in my bio. All credit goes to the authors for all findings of this paper, and I want to thank them personally for allowing their work to be shared.